few months. It's a larger team, so it's a team of 12, and they're entering this international competition in October, which isn't sports specific, it's sort of a general science competition. So I'm not going to say too much apart from a couple of organisational things, which is that they'll talk for about 25 minutes or so, and then we'll have time for questions. And then I'm aware, obviously I've said you have the room booked till 4 o'clock, and maybe not everyone can stay that long, so feel free to leave if you have to at any stage. But what we're kind of hoping is that maybe in the second half of that time, if people can stay, we'll have a little bit more of a kind of close examination and maybe a little bit of sort of group work on some of the issues that are raised, because the whole purpose of them coming, in a sense, is just obviously to explain their project, but also to get feedback on how this might actually work in real life, how sports organisations would respond, how athletes would respond. I'm going to stop talking, otherwise I'll be here for 25 minutes. So I'll hand over to you. Thanks very much.
Thank you very much, Paul, for introducing us so nicely. Once again, my name is Jard and this is Gemma, and we're from Delft University of Technology in the Netherlands. And we are participating in the International Genetically Engineered Machine Competition in Boston at the end of October. And as part of this, we are developing a method for detecting gene doping under the name Advanced Detection of Performance Enhancement Dope. So a bit more about today. As Paul has already mentioned, first we'll present our project. After this, there is some time for questions from your side, and after we would like to engage in a discussion with you on how gene doping is different from currently more conventional methods of doping in the sense of its impact on environment and society, as well as its doping development cycle. But first, of course, let me set the case for you about doping. Of course, you're all familiar with it, and doping is very often all over the news. For example, with this newspaper article from Sochi, where at least 13 Russian athletes had to hand in their medals afterwards because they used doping. So it's really happening a lot. And what about this guy, Lance Armstrong, who was able to win the Tour de France 7 times using doping? It's really the news. Everyone was shocked. So what if there is this new kind of a relatively new kind of doping? And in 2004 already, there was a research group at Howard Hughes Medical Institute in the US who were able to engineer marathon mice that could actually run twice as far as normal mice just by changing one gene coding for one protein, which was really remarkable. And 2 years later, only in 2006, there was a coach, a German coach, Thomas Springstein, who was convicted for trying to get his underage athletes on a new kind of gene therapy used for performance enhancement. So from this year on, the World Anti-Doping Agency actually announced that they want to take active measures against this type of doping, this gene doping. And this is also where we come in, because there have been several methods already developed. But there are several values at the core of our project, and those are listed here. We want to have a method that is efficient in the sense that we do not want to process whole genomes. So it's actually a method in which it can be easily implemented by many organizations. Also, we want to take into account athlete privacy to a large extent. So we don't have to sequence their whole genome in order to be able to tell whether they use gene doping or not. And then lastly, in gene doping, there is a plethora of possibilities of the possible changes that can be taken into account to actually enhance performance. So we do want to have a method that is not only specific for one type of gene doping, but for all of them. So that's why we came up with our method that we'll come to talk about more extensively later. But why is this important? Because we often get the question, why is gene doping actually happening? Is this really a thing? And we, of course, it's really hard to talk to athletes about doping and whether they use doping. No one will say, oh, I use doping or I use gene doping, if you interview them, or well, at least not many. So we did an assessment like across society also. We had these debates both in the Netherlands as well as in China, and we asked people, just general people in the streets, would you like to use gene doping for performance enhancement in any field? And in the Netherlands 15% actually said they would like to do this. Whilst in China it was 55% that actually is interested in enhancing their performance. And these actually, even the 15% is actually a huge amount, and given the pressure that is on athletes, we expect that these figures are even higher in this athlete population. When we were in one of our debates, we also talked to an athlete, and he said, if it's undetectable and if it's safe, then everyone will be using this, so why not me? This is really the idea that they have in mind at present. But first, like for the ones of you who are not very familiar with how gene doping works, you here see an athlete, and actually the idea is that they can inject the vectors, so-called, into their bloodstream or into their muscles. And these vectors can either be several types of viruses, most likely at present the adenoviruses, so they can be controlled a bit better, but also like plasmid DNA, which is a small circular DNA, which is normally found in bacteria. And both in these viruses as well as in these plasmids, you can insert the gene of interest, which could be EPO, as the general doping gene, but could also be any other gene that is associated with performance enhancement. So they can inject this, and actually don't be scared or afraid. We modeled this, actually what happens after injection. So we looked at the EPO concentration in the blood, and that's what you see in black. You see the spike after injection at the time 50 days. And this spike actually then results later on through a whole complete process in the physiological process into a spike of the red blood cells later on, where these are the red ones and the black striped ones are the reference levels. So you do see that there is a clear impact after the injection of the vectors. So the way it actually works on a physiological level is they inject the vectors, they can get into your bloodstream, into your tissue, and in your tissue they can enter cells, and in the kidneys, for example, if we talk about EPO, it can enter interstitial fibroblasts, as they are called, and those are the main cell population responsible for the production of EPO, not the only one though, but the main one. And from there, EPO will get into the blood, and it will be distributed, it will be having an effect then on the bone marrow in the red blood cell production. And this I can show you here in a simplified figure. In the bone marrow there are pluripotent stem cells, so those are cells that can differentiate into all types of blood cells. And then after they specialize, then you can see a few E cells where erythropoietin has a direct effect on the maturation time, as well as for the proerythroblasts, and after this it has a direct effect on the time control and the time that the reticulocytes will stay in the bone marrow before they will be released and become red blood cells. So actually this is the effect of EPO. It actually controls the time that it takes from the stem cells to mature into the red blood cells in the blood. So then this is what you actually see as the red peak, as I told. You see the red blood cell increase, which is actually approximately 50 to 150 days after the injection of the vectors. So the idea is that athletes, if they want to have this peak around competition time, they would dope or gene dope in between any competition. So this gives us our time frame for detection. So we would have to detect not only before the competition, but especially in between competitions. That is what it says. And of course, if the red blood cells are in the blood, they increase the oxygen transportation and thereby may enhance the performance of an athlete. And you have this regulatory feedback loop, of course, but with messing around, you could enhance the oxygen transport, which can enhance performance, but which can also be dangerous if you really mess around with this feedback loop, causing strokes, for example. So what do we actually do? Because we've been talking about EPO and the regulation circle and how you can interfere with this. But our idea is we want to detect gene doping. And this is what you see here. You see that from the cell, these cells, they can die, of course, in a time span, and then they will release their DNA partly. And this will form cell-free DNA. It's also the vectors that are not uptaken into the tissue and into the cells, those also will form the cell-free DNA, and that is actually what we want to target. So in the graph you also see what we estimated to be our detection window, which is approximately 17 days, which is actually corresponding to what we found in literature on primate research. And it's actually not even that short compared to some other substances that the doping authorities are currently looking for, based on a talk we had with the Dutch doping authority. So we're quite hopeful on that side. So what we do in our method is we take blood samples from athletes, and then we have an extensive sample preparation method to extract the DNA from these samples, and that's what we want to target for. But of course we now have been again looking for this DNA that codes for EPO, but this DNA could also code for several other genes that are involved in performance enhancement. For example, myostatin blockers, which you see in the cow over there. It has a huge effect on the muscle mass, but also they've been testing this type of doping on rabbits, for example, where they saw that the tongue growth actually increased, which also had lethal effects on those rabbits. So you see that there is a very hard time predicting what the actual effect of this gene doping will be, and especially when you transfer it then to humans, you just don't know based on those animal studies. Then there are several other factors like growth hormone and vascular endothelial growth factor that promotes the vascular growth, and then ACTN3, which is involved in skeletal muscle development, but also, for example, endorphin, which can relieve the athlete's pain barrier. This is a completely different angle. But then, again, what are we doing? And this is something that Gemma will talk a bit more about.
Yeah, thank you. I think I'll have to walk that way.
Yeah.
Yeah, so thank you, Jörn. So I want to outline for you what's actually the method that we have developed. And it comprises of two individual tests. One is a pre-screen in which we're able to fast rule out most of the samples. And the other one is a method we have developed ourselves fully, which is a method based on sequence verification. So the output of that final method is actually the sequence that the athlete has used to gene dope, which makes it a very good proof that an athlete has actually used gene dope, and we also have some more insight on what exactly this person did. So the first thing that I actually want to talk to you about is what we are actually targeting, because we are targeting DNA in the blood, and we can see with very subtle differences the difference between doping DNA and natural endogenous DNA. So here the top bar you see endogenous DNA from the human body as it is naturally, and below you see our doping gene. And what you can often see in what we expect to see in doping DNA is that the non-coding parts of the DNA will have been cut out because it is easier to transfect with a smaller vector, so with less DNA, than it is with more DNA. So to increase the stability and the efficiency of the gene dope, of the method of gene doping, we expect that many cheating athletes will actually remove the non-coding here in white DNA parts. And then where we're actually targeting is these junctions, as you see here at the arrows, between the blue and the blue part. They're called exon-exon junctions because those are not occurring in nature. So the first test we do is a pre-screen. So this test is based on dextrose gold nanoparticles, which was a method first developed by Anderson et al. At Michigan State University. So it's based on color change due to stability of gold nanoparticles. So you see here the DNA over here, that's our target DNA, which is in red the actual doped DNA, the gold nanoparticles, and a template DNA strand that's complementary to the target DNA. So when the template DNA binds to the target DNA, it gives the gold nanoparticles space to also bind to the DNA, which stabilizes them. When you then add salt to the solution, the stable gold nanoparticles make sure that the color change occurs slowly. So when you have when you add normally to free gold nanoparticles, you add a salt solution, it turns purple faster than when you do this to the stabilized gold nanoparticles. So in this way, by comparing our athlete samples to a reference sample, we are able to see with a slow color change if a person has used gene doping or not. So basically, if it stays red for longer, this person is a very likely subject to gene doping. So when we flag the sample this way, we move on to our fusion protein method. So the fusion protein, we have described it here, is based on a Cas. Many of you maybe know it from the CRISPR-Cas system. This dead Cas is able to read the DNA and bind to a target. It is fused with the linker over here to a transposase. The transposase is able to cut the DNA and prepare it for further sequencing analysis. You can see here that there's another Cas protein. This one we don't actually, it's not actually functional, but due to the way we make the fusion protein, most often the double Cas will be attached instead of the single Cas. But this is not a problem for our detection method. So I'll go talk you through the method one by one. So first thing that happens is that the Cas protein scans the DNA and finds the target. As you can see here, the red RNA it has inside of it matches the red DNA that's in our target, and then it binds to it. When it's bound, this gives a fusion protein, which is in very close proximity to our site, the chance to cut the DNA and add the adapter sequences it has carrying with them. So here you see that the transposase has cut the DNA and has added the blue sequences it was carrying to our DNA. These blue sequences, we call them adapter sequences. They're necessary for the sequencing analysis, but we can also add them to add all kinds of genetics information about the sample. So for instance, the name of the athletes, the time and date of the sample collection, things that might be useful and might reduce tinkering with samples. So when the transposase has cut and added the adapter sequences, it lets go of the sequence again, and this allows the motor protein here in purple to find the blue sequences. So it's tagged to find the blue sequences. And then the motor protein pulls our DNA, which we want to sequence, through a nanopore. The nanopore is part of the sequencing platform developed by Oxford Nanopore Technologies. It's based on an electric current that goes through this nanoparticle, and because of disturbances in the current every time a base passes through, we're able to read out the sequence in this way. So the Cas will fall off, the fusion protein will fall off as it's being pulled through, and then we get a very reliable readout of the sequence. So this is some results of what we've done in the lab thus far. So the first one you see is the transmission electron microscope image of these dextrin-capped gold nanoparticles. You can see we here test their functionality, and we have seen that they're 13 nanometers in diameter. And below you see one of the reference samples where we used the saline solution to see the color change. So I'm not sure if it's super visible, but you can see it starts red and it ends up purple over here. The one next to it is a protein gel in which we've purified our fusion protein. So you see here in the 5th lane, it's our fusion protein at the expected height. So as we've been developing our method, we've asked for help from many different people to understand the problem better and to make sure that our method is actually helping with all needs. And as we've been talking to, for instance, the Dutch National Blood Bank, we've discovered that there's many other applications that a targeted sequencing, as we're developing, could actually help with. One of them is non-invasive prenatal screening, in which you could take the blood sample from the mother's DNA instead of having to probe from the child. Another one is, for instance, bacterial identification, in which you could look at very complex ecosystems, in, for instance, soil, to find trace elements of bacterial DNA that we would have missed otherwise. So a slight look into the future of our method. So we're working until the competition in October to develop our first proof of concept. So we're right now linking all parts of our method together to try and get a first functional proof of concept based on EPO and the exon-exon junctions. And after that, the lab that we're currently under supervision of, the Stan Brown's lab, is going to apply for a grant to continue this process in the future, most likely with some of our student members currently also at work. So the future of our method is very much dependent on this grant application. So before we close, I'd very much like to thank all our sponsors. Our research is very much based on the money that we are able to get from many private and public instances. So without them, it would not have been possible. And also our amazing teammates back in the Netherlands working very hard on our method as we speak. Our amazing team of supervisors that help us so much, and also many others that we have talked to and that have helped guide us in the development of our method. So we'd now be very much open to all your questions.
Okay.